What is the DNA? It stands for deoxyribonucleic acid. It's the genetic code that determines the characteristics and features of all living thing. We get our DNA from your parents, we call genes, and their positive charges allow them to associate with DNA, which is negatively charged. Some histones function as spools for the thread-like DNA to wrap around. Under the microscope in its extended form, chromatin looks like beads on a string. The beads are called nucleosomes. Each nucleosome is made of DNA wrapped around 8 histone proteins that function like a spool and are called a histone octamer. Each histone octamer is composed of 2 copies each of the histone proteins H2A, H2B, H3, and H4. The chain of nucleosomes is then wrapped into a 30 nanometers spiral called a solenoid, where additional H1 histone proteins are associated with each nucleosome to maintain the chromosome structure. Transcription. Transcription is the process by which DNA is copied, transcribed, to mRNA, which carries the information needed for protein synthesis. Transcription takes place in 2 broad steps. First, pre-messenger RNA is formed, with the involvement of RNA polymerase enzyme. DNA, RNA and protein synthesis. The genetic material is stored in the form of DNA in most organisms. In humans, the nucleus of each cell contains 3 times 109 base pairs of DNA distributed over 23 pairs of chromosomes, and each cell has 2 copies of the genetic material. This is known collectively as the human genome. The human genome contains around 30,000 genes, each of which codes for 1 protein. Large stretches of DNA in the human genome are transcribed but do not code for proteins. These regions are called introns and make up around 95% of the genome. The nucleotide sequence of the human genome is now known to a reasonable degree of accuracy, but we do not yet understand why so much of it is non-coding. Some of this non-coding DNA controls gene expression, but the purpose of much of it is not yet understood. This is a fascinating subject that is certain to advance rapidly over the next few years. DNA replication. Each time a cell divides, each of its double strands of DNA splits into 2 single strands. Each of these single strands acts as a template for a new strand of complementary DNA. As a result, each new cell has its own complete genome. This process is known as DNA replication. Replication is controlled by the Watson-Crick pairing of the bases in the template strand with incoming deoxynucleotide triphosphates, and is directed by DNA polymerase enzymes. It is a complex process, particularly in eukaryotes, involving an array of enzymes. Chromosomal mutation. Microevolution is based on the changes at a molecular level that cause species to change over time. These changes may be mutations in DNA, or they could be mistakes that happen during mitosis or meiosis in relation to the chromosomes. If the chromosomes are not split correctly, there may be mutations that affect the entire genetic makeup of the cells. 1. Chromosome duplication. Since sister chromatids are exact copies of each other, if they do not split down the middle, then some genes are duplicated on the chromosome. As the sister chromatids are pulled into different cells, the cell with the duplicated genes will produce more proteins and overexpress the trait. The other gamete that does not have that gene may be fatal. 02. Of 04. 2. Chromosome deletion. If a mistake is made during meiosis that causes part of a chromosome to break off and become lost, this is called a deletion. If the deletion occurs within a gene that is vital for the survival of an individual, it could cause serious problems and even death for a zygote made from that gamete with the deletion. Other times, the part of the chromosome that is lost does not cause fatality for the offspring. This type of deletion changes the available traits in the gene pool. Sometimes the adaptations are advantageous and will become positively selected for during natural selection. Other times, these deletions actually make the offspring weaker and they will die off before they can reproduce and pass the new gene set down to the next generation. 3. Chromosome translocation. When a piece of a chromosome breaks off, it is not always lost completely. Sometimes a piece of chromosome will attach onto a different non-homologous chromosome that has also lost a piece. This type of chromosome mutation is called translocation. 4. Chromosome inversion. Another option for a piece of chromosome that has been broken off is called inversion. During inversion, the piece of the chromosome flips around and becomes reattached to the rest of the chromosome, but upside down. Unless the genes need to be regulated by other genes via direct contact, inversions are not as serious and often keep the chromosome working properly. If there is no effect on the species, the inversion is considered a silent mutation. Motor neuron disease and its correlation between a dormant retrovirus carried in the ancestor's DNA. Way back in time, ancient viruses are believed to have infected the reproductive cells of our ancestors, leaving behind fossils of their genomes that we still see in our DNA today. In fact, as much as 8% of our DNA is made up of the remnants of these infections, known as endogenous retroviruses. Usually they lie dormant, but they can be awoken, reigniting the production of viral elements. Controversially, such reactivation of these sleepy sequences has now been linked with a progressive disease of the nervous system, called Lou Gehrig's disease. We're way off from confirming that these viral genes play a causative role in this condition, but if this turns out to be the case, then it may be that some patients can be helped with drugs that we already use to treat infections with similar viruses. This finding followed a series of observations regarding the condition, also called amyotrophic lateral chromosomal mutation by way of deletion. In genetics, a deletion, also called gene deletion, deficiency, or deletion mutation, is a mutation, a genetic aberration, in which a part of a chromosome or a sequence of DNA is lost during DNA replication. This uncopied part can be as small as a single nucleotide or as much as an entire chromosome. The loss of this DNA during replication can lead to a genetic disease. Diseases that can be caused by deletion mutation can include 22q11.2 deletion syndrome, cystic fibrosis, Turner syndrome, and Williams syndrome. 22q11.2 deletion syndrome, which is also known by several other names, listed below, is a disorder caused by the deletion of a small piece of chromosome 22. Many children with 22q11.2 deletion syndrome have developmental delays, including delayed growth and speech development, and learning disabilities. Chromosome duplication. A type of mutation in which a portion of a genetic material or a chromosome is duplicated or replicated, resulting in multiple copies of that region. Supplement. Duplication results from an unequal crossing over between misaligned homologous chromosomes during meiosis. Gene duplications are an essential source of genetic novelty that can lead to evolutionary innovation. Duplication creates genetic redundancy, where the second copy of the gene is often free from selective pressure, that is, mutations of it have no deleterious effects to its host organism. Duplication mutation. A type of mutation in which a portion of a genetic material or a chromosome is duplicated or replicated, resulting in multiple copies of that region. Duplication results from an unequal crossing over between misaligned homologous chromosomes during meiosis. Gene duplication is the process by which a region of DNA coding for a gene creates additional copies of the gene. Similar to gene duplication, whole genome duplication is the process by which an organism's entire genetic information is copied, once or multiple times, which is known as polyploidy. Chromosome inversions. An inversion is a chromosome rearrangement in which a segment of a chromosome is reversed end to end. An inversion occurs when a single chromosome undergoes breakage and rearrangement within itself. Inversions are of two types, paracentric and pericentric. In some cases, crossing over is suppressed. During meiosis, homologous chromosomes are paired. In other paracentric inversions, crossing over can occur, but the products of crossing over are never recovered because the resulting chromosomes are non-functional. An inversion is a chromosome rearrangement in which a segment of a chromosome is reversed end to end. An inversion occurs when a single chromosome undergoes breakage and rearrangement within itself. Inversions are of two types, paracentric and pericentric. Chromosome translocations. Translocations generate novel chromosomes. In a translocation, a segment from one chromosome is transferred to a non-homologous chromosome or to a new site on the same chromosome. Translocations place genes in new linkage relationships and generate chromosomes without normal pairing partners. When scientists compare the genomes of closely related species, they can see that translocations have occurred many times during the course of evolution. However, translocations that give an organism an adaptive advantage are very rare. Translocations are more often associated with negative consequences such as aneuploidy, infertility, or cancer. Scientists are now able to use molecular probes to analyze translocations, and the results are providing new insights into the molecular origins of various diseases, including cancer. Translocations generate novel chromosomes. Translocations were first detected cytologically in the late 19th and early 20th centuries as novel chromosomes that appeared prominently in tumor cells. Some of the earliest, most complete descriptions of tumor cell chromosomes were provided by the great German cytologist Theodor Boveri, considered by many to be first cancer geneticist. Based on his observations of tumors, Boveri postulated that tumor cells possessed growth stimulatory chromosomes that played a role in malignancy. Baumann, 2001. At the time, cytological markers were not available for human chromosomes, so Boveri was not able to identify specific chromosomal changes in tumors. Today, we know that Boveri's insights were correct. Translocations are indeed common in cancer cells, and some translocations produce oncogenes that are responsible for malignant transformation. DNA repair defects in cancer mammalian cells employ at least 8 distinct DNA repair pathways to cope with a multitude of different genotoxic lesions. This network of genome maintenance pathways consists of mismatch repair, MMR, base excision repair, BER, nucleotide excision repair, NER, translesion synthesis, TLS, homologous recombination, HR, non-homologous end joining, NHEJ, and the Fanconi anemia, FA, and the O6-methylguanine DNA methyltransferase, MGMT, pathways, 12,19 to 41. MMR recognizes erroneous insertions, deletions, and misincorporations of bases that have been neglected by the intrinsic proofreading activity of the DNA polymerases. These lesions are detected by the MITSA complex, detection of small mismatches, formed by MSH2/MSH6 and the MITSB complex, detection of large mismatches and insertion loops, formed by MSH2/MSH3. In eukaryotes it was proposed that the lagging strand transiently contains nicks that are coded by the B-sliding clamp pCNA and a replication factor C, RFC-dependent man-to-mutlock complex, formed by MLH1 and PMS2, connects much with the pCNA/RFC complex by a mechanism that is not fully understood. Upon binding, the exonuclease XO1 is recruited to the MUTS/mutlock complex and the lesion gap is filled by DNA PO. Single-stranded breaks, SSBs, and non-helix distorting base modifications are repaired by BER. Modified bases are detected and excised by DNA glycosylases, generating an obesix site. Part 1, see glossary, and 2 are important sensors for SSBs and facilitate recruitment of additional BER factors such as XRCC1 to the site of genotoxic injury. Subsequently, XRCC1 recruits the APE1 nuclease, which cleaves the obesix site at the 50 position. Next, RFC recruits a complex of pCNA and DNA PO, E to the damaged site, and this displaces and resynthesizes 2 to 8-NT from the damaged site. Finally, the endonuclease FEN1 cleaves the dislodged oligonucleotide and the SSB is resealed by ligase I. Helix-distorting lesions, which are induced by UV irradiation and platinum-based chemotherapeutics, are repaired by NER. NER can be initiated by global genome NER, GG-NER, recruiting primarily XPC/RAD23B and RPA/XPA complexes to the damaged site or by transcription-coupled NER, TCNA, recruiting a complex of XPG and CSP to sites of RNA polymerase stalling. GG-NER and TC-NER culminate in recruitment of TFI. This factor incorporates the helicases XBB and XBD and unwinds a 30 NT fragment around the damaged site. After DNA unwinding, the complexes XPF/ERCC1 and XBG are recruited, and these exhibit nuclease activity at the 50 and 30 ends of the lesion. Finally, the damaged site is resynthesized by complexes consisting of DNA PO, E, RFC, and pCNA or, alternatively, DNA PO, E and XRCC1, 49. The SSB is sealed by ligase or IEA. Mammalian cells employ two distinct pathways for DSB repair, HR and hedge. HR is an error-free DSB repair pathway that is largely restricted to the SNG2 phases of the cell cycle, when an intact sister chromatid is available as a template. Next, RFC recruits a complex of pCNA and DNA PO, E to the damaged site, and this displaces and resynthesizes 2 to 8 NT from the damaged site. Finally, the endonuclease FEN1 cleaves the dislodged oligonucleotide and the SSB is resealed by ligase. Helix distorting lesions, which are induced by UV irradiation and platinum-based chemotherapeutics, are repaired by NIR, Figure 1c. NIR can be initiated by global genome NIR, GG-NIR, recruiting primarily XPC/RAD23B and RPA/XPA complexes to the damaged site, 40, or by transcription-coupled NIR, TCNOL, recruiting a complex of XPG and CSB to sites of RNA polymerase stalling. GG-NIR and TC-NIR culminate in recruitment of TFI. This factor incorporates the helicases XBB and XBD and unwinds a 30 NT fragment around the damaged site. After DNA unwinding, the complexes XPF/ERCC1 and XBG are recruited, and these exhibit nuclease activity at the 50 and 30 ends of the lesion. Finally, the damaged site is resynthesized by complexes consisting of DNA PO, E, RFC, and pCNA or, alternatively, DNA PO, E and XRCC1. The SSB is sealed by ligase or IEA. Mammalian cells employ two distinct pathways for DSB repair, HR and hedge. HR is an error-free DSB repair pathway that is largely restricted to the SNG2 phases of the cell cycle, when an intact sister chromatid is available as a template, 50, Figure S1. Upon initiation of the HR process, the DSB is resected to create a single S-stranded, SS30 overhang, which becomes rapidly coated with the SNA-binding protein RPA. This SNA overhang ultimately invades the sister chromatid. Once an RPA-coated SNA filament is generated, RPA is replaced by RAD51 in an ATM/CHK2/BRCA1/BRCA2/PALB2 dependent fashion. RAD51 lies at the heart of the HR mechanism and mediates homology search, strand exchange, and holiday junction formation. The enormous importance of the HR pathway for genome maintenance and cancer prevention is underscored by the observation that patients with heterozygous germline mutations in different HR genes, such as BRCA1, BRCA2, and RAD51C, display a dramatically elevated risk for the development of cancer. Furthermore, disabling mutations in BRCA1, BRCA2, ATM, CHEK2, RAD50, RAD51C, and others have been recurrently identified in numerous cancers, including lung cancer, ovarian carcinomas, pancreatic duct carcinoma, and chronic lymphocytic leukemia, CLL. In addition to HR, mammalian cells also employ the NHEDGE pathway to cope with DSBs. NHEDGE-mediated DSB repair does not require the presence of an intact template. Multiple sclerosis ALS or motor neuron disease, which slowly destroys the nerves involved in movements. While roughly 10% of ALS patients also have a relative afflicted with the disease, indicating heritability, some have no family history, hence the name sporadic ALS. Interestingly, some patients with AIDS, caused by the retrovirus HIV- show symptoms reminiscent of ALS, but they can often be reversed using antiviral drugs. Tenuous to some, but another hint of a possible connection came from blood samples of ALS patients, which in some cases showed the presence of a retroviral protein. reverse transcriptase, that is used to turn their RNA genome into DNA. Scientific studies which examined brain tissue to look for viral signatures revealed that one particular endogenous retrovirus, called HERV-K, was activated in the samples from deceased patients with sporadic ALS, but not healthy controls. By activation, the scientists are referring to the genes being made into blueprints for proteins, which are called transcripts.